Now we discuss about special modes of reproduction. Number 1, apomixis. In this process, new organisms are developed without any fertilization. It is of two types, vegetative and agamospermic. What is about vegetative reproduction? In this process, only vegetative parts are involved. In this process, the vegetative parts which are derived from a sporophytic plant like bulbils, gamules, etc. But what is agamospermy? In this case, embryo is developed without any fertilization. It is of two types, embryonic and gametophytic. In case of embryonic, one or more diploid cells are involved or sporophytic cells are involved in the development of embryo. In case of gametophytic, in this case, embryo is developed from megaspore mother cell or near adjacent nucellar cells. It is of two types, diplospory and apospory. In case of diplospory, the embryo is developed by the gradual development of megaspore mother cell. But in case of apospory, embryo is developed from adjacent nucellar cells which behaves as megaspore mother cell. Then second one is the parthenogenesis. It is one type of special modes of reproduction. In this case, embryo is developed without any fertilization. It occurs both in plants and animals. It is of two types, diploid and haploid. In case of diploid parthenogenesis, it undergoes both diplospory and apospory case. Already we discussed about diplospory and apospory. In both of the cases, both diploid embryo is developed. So it is called as diploid parthenogenesis. But in case of haploid parthenogenesis, in some cases, male gametes fail to fertilize with egg cell. In this case, this haploid egg cell develops an haploid embryo. So this process is called as haploid parthenogenesis. Then third one is the parthenocarpy. In this process, seedless fruits are developed. It is only applicable for plants. It causes through many processes. Number 1, genetic parthenocarpy. In this case, it occurs some due to some genetic disorder. Then second one is the due to environmental changes. Then third one is the chemically induced parthenocarpy. In this case, some chemicals causes seedless fruits. Chemicals like auxin, gibberellin, etc. Then fourth one is the polyembryony. This process was first discovered by Antony von Leeuwenhoek. In this process, more than one embryos are developed inside a single seed. It is of two types, true and false. In case of true polyembryony, these embryos are fertile. But in case of false polyembryony, the embryos are sterile. Now we discuss about true polyembryony. In this case, but this polyembryony process occurs through many causes, through many purposes. Like first one is the single embryo sac contains many egg cells. Second one is the zygote divides into fragments. Third one is the synergid cells acts as egg cells, etc. But in case of false polyembryony, in this case, a number of embryos are formed due to multiple embryo sacs. But these embryos are sterile. The last one is the tissue culture. It was first proposed by Stewart. This process totally based on totipotency. That means the total potentiality of a plant cell to develop a whole plant. Its main purpose is to develop more clones within short period of time. It only occurs inside the well-developed laboratory. Thank you.